Well, I do believe that we have two major issues that we need to be in some way addressed in the diagnostic workflow for MTB. The first one is to achieve a larger sensitivity in the method that are currently used, like PCR and culture and microscopy. And the second one is to be able to detect in a very fast way the presence of resistance gene against a specific therapy. On the other hand, we still have issue with the performance of the IGRA test, so the interferon gamma related assay. And because most of the tests that are already on the market are working in a good way, but sometimes we got a quite high percentage of undetermined test. That means that we need to recall the patient, repeat the test. That's a major issue because it's quite a complicated way. So we spend a lot of effort in solving this. So we need to improve this tool technology. Well, I think that the era of multiplex PCR panels is well developed right now, and my personal feeling is that this will be over in about 5 years. So we will move to a different perspective. That's the one of NGS sequencing, either shotgun or metagenomic or whole genome sequencing. But this will be a different approach, completely different approach. Of course, you need to have two relevant basic points when you try to apply NGS in diagnostics. The first one is that you need to have a very good pre-analytical automation that can ensure the perfect identification of the sample during the entire workflow. Otherwise, it will be a mess. And this is just not only useful for the identification of the sample, but also to reduce the time that is required. So my laboratory will switch from manual preparation to robotic preparation, and this means that we can go for about 80 samples in 8 hours. So that's a very good time. And then, of course, the second step is to improve the quality of the sequencers that you have. The very last sequencers that are available can reduce from 16 to 7 hours the reading time. So taken together, this at the very end means that you can provide results within 36 to 48 hours from the arrival of the sample in the laboratory. That means it's well comparable with the standard culture time of bacteria, and it makes a lot of difference because you are much more effective. You are not sticky to what is capable to grow. Question mark point is that you need to explain very careful to the clinician what is the meaning of what you are reporting. Because if I report the Staphylococcus aureus, it's clear for everyone what should they do. But if I'm reporting a strange bug or a strange fungus, well, we need to discuss with them the relevance, the possible relevance at the clinical point of view. But I'm really convinced that in the next 5 years, about 50% of what will be reported will be generated by NGS. So I think that will be our future. Well, I'm following the IVD company coming from China since, I would say, almost 10 years. And I saw them grow a lot in terms of technological capacity and in terms of scientific results. So I believe that they are now very open and very ready to go onto the market in Europe. So I think that what we are expecting from them, and they are showing that for this type of company, this is possible, is to have a high flexibility in responding to the customer request and to, at the very end, reduce the price of what we pay for diagnostic. And that will be a major, possibility for them to enter into the European market. I do believe that we are starting to speak the same language. So that means that we can get in touch, get in connection, and discuss openly whatever it's a need we have and whatever is a possible need that this company has. Because what I believe we can provide them is a different approach, because the European regulation is very different from the one we have in Asia or in other parts of the world. So first of all, they need to understand how to manage their relationship with us as public laboratory located in Italy or in Europe. And on the second step, I would say that the possibility for them to enlarge the portfolio of data that can be used in order to verify or assess the validity of their techniques, it's huge because, of course, especially in infectious disease, the prevalence of different diseases is not the same in Europe and in Asia. And the normality value are not the same. So I think that they need to get into the European market, trying to establish a good collaboration and to collect data that can be used to get into the market in Europe, but the data should be collected in Europe. That makes a lot of difference because it will be a much more trustable situation if they collect data in Europe. But I see a huge possibility for them to collaborate. Well, I'm a physician by training, so I would like to have physicians following my steps, so working into the laboratory, because I believe that nowadays we have a lot of people that is very well trained under the technological point of view, like biotechnician or biologist, but we need to speak the same language with the clinician, because other way it will be, a kind of parallel worlds that will not get in a useful touch together. And so I think that we do need to have some more young physicians to get there and to work in strict connection with the biotechnologists that can develop new tests and apply that. That will be the right combination. If we leave the laboratory in the hand of biotechnologists, that will be a major issue because clinicians will try to drive the laboratory. And that's not good because the laboratory nowadays is a very complicated machine. So we do need to have people inside the laboratory saying, okay, I know how it works, and I know how to give you in the clinical side the result you need, but it's not possible for the clinician to jump into the laboratory and drive the laboratory because they have the right, training, the right scientific training. And so that will be probably the best opportunity for young people to stay into the laboratory and to open their mind and to their voice to the clinician side.